Hi everyone. So today's lecture comes from your PowerPoint that's labeled Chapter 10. We're going to talk about the origin and diversification of life on Earth. So this is really cool. I love this image because this is an image of the feet of lizards. And as you know, they can climb walls. If you've ever had a gecko or lived in a place where there are geckos, you know they can go straight up the wall. And it's because of these specialized pads that they have. And actually, startups and scientists have looked at the formation of these pads in order to make specialized gloves so that we too can climb walls. So, very cool. I want to tell you about an experiment that was done quite some time ago that everyone who takes biology needs to know. This is the Urey-Miller experiment, and it's actually pretty fascinating. And so what Urey and Miller, they were two scientists who worked on this experiment, they wanted to know whether or not they could create the substances of life from what was thought to exist on early Earth. Right? When you think back to, all that we have learned and we know about early Earth, where there's one theory that it was very hot, right? And you had these electrical storms going on, and you had this, quote, primordial ooze that was there. So the question is, well, how did all of this diversity come out of that? How did life come out of that? And so in this experiment, what they wanted to try to determine is whether or not they could recreate early Earth and see whether or not they could get some of the products of life. So here's what they did. So they took a flask, they were chemists, they took a flask and they filled it with what has been thought to be on early Earth, hydrogen, methane, and ammonia, and they put it in the flask. And because it was thought that early Earth was hot, they heated it up. And then this was simulating the atmosphere that was present in early Earth, right? And then to simulate the electrical storms that were going on, they had an electrical discharge that was in a separate flask. So once you heat it up, then you have the atmosphere, which they created, going into this separate flask, which received an electrical charge. And then they cooled it down, because that's what is thought happened, because of course the Earth is not boiling right now. So they cooled it down, and then they collected the condensates, what came out of that cooling. And here's what's interesting. Within a matter of days, they discovered many organic molecules, including 5 different amino acids in their primordial sea, in what was in this condensate. And so that was sort of mind-blowing and game-changing, that you could put hydrogen, methane, and ammonia and heat it up and then send an electrical charge through it, then cool it down, and out pops amino acids. And so this was, of course, repeated over and over, and it was repeated recently, and all 20 amino acids were actually found. And they went back and pulled the samples, the early samples from Urey and Miller's experiment, because now, we have much more sophisticated equipment to analyze it. And when they reanalyzed their experiment, they also found more amino acids in there. So click chemistry is something that is certainly possible and could explain the life that we see on the planet. And so when we start looking at the timeline, remember when we were looking at evolution, and before Darwin, they thought the Earth was only 6,000 years old. Well, we have found fossils of cells, early cells, what we feel is the earliest life on Earth, fossils of 3.4 billion with a B, 3.4 billion year old cells that may have been among the first living organisms on Earth. So let's take a look at it as we in science. So it's all about what we can measure, right? What the data says. And so when we question, because that's what science does, you constantly question, like, how did this happen? How could this happen? Let's create an experiment, much like Urey and Miller, and then measure and analyze the data. And so when we question how did the first organisms on Earth arise, so many scientists believe that life originated on Earth probably in several distinct phases. So first you had the formation of small molecules containing carbon and hydrogen. Because of carbon's chemical structure, remember we have those 4 open valence spaces. Because of carbon's chemical structure, it can form a huge variety of molecules with wide and varying functions. And then you have this formation of self-replicating information-containing molecules. And so the nucleic acid RNA can both carry information and catalyze the reactions necessary for replication. Thus, this single molecule could have been a self-replicating system and the precursor to cellular life. And it turns out that when scientists replicated a different version of Urey Miller's experiment, out popped RNA. So it's certainly possible, because just by doing those very, very simple steps, replicating early Earth, you get amino acids and you get RNA basically coming together like click chemistry. And then, of course, what has to happen is you have to have the development of a membrane. And so the membranes compartmentalize the self-replicating molecules, the DNA, the RNA, from their surroundings, facilitated by metabolic activity. And we know that membranes will also self-organize when we look at our phospholipids. Remember, they have the hydrophilic and the hydrophobic portions. You put them in water, click chemistry, they are going to form these membranes. So very, very interesting. But that takes us to all the diversity that we see on the planet, right? And so here, when we take a look at these 2 individuals, we easily recognize Cristiano Ronaldo of Madrid and Beyonce Knowles as members of the same species, right? They're Homo sapiens. But when we look at other organisms, for example, the Persian buttercup and the pink rose, or even when we look at the Bornean orangutan and the golden lion tamarin, they can look very similar. However, they are not from the same species. So this begs the question, what is a species, right? So here is your definition of what is a species. Species are, number 1, populations of organisms that interbreed with each other or could possibly breed under natural conditions. Now, under natural conditions is that really super important disclaimer that's really important. So it means not, us, the scientists, creating this new organism, because that's not under natural conditions, right? So it has to be that they can interbreed with each other or could possibly breed under natural conditions, and they have to be reproductively isolated from other such groups. That is your definition of a species. So when we look at something, this organism, which we call a zorse, this is half zebra, half horse. So even though we can produce them in captivity, organisms that are not naturally occurring in the wild, like such as this offspring of a zebra and a horse, they are not considered a species because we created them. A horse and a zebra are not going to naturally breed in the wild. Okay, so not considered a species. So when we look at the reproductive isolation, what is keeping species separate? Why can't other organisms interbreed with other organisms, right? That's a pretty valid question. Well, we've got barriers. Nature has barriers. So there's a pre-zygotic barrier before the zygote, right? That makes individuals physically unable to mate with each other. Or if they're actually able to mate, the male's reproductive cell is unable to fertilize the female's reproductive cell. So that's a pre-zygotic barrier. Can't even become a zygote, right? They're either not physically able to, as we can see here, we compare this pig with a dog, just not physically possible. Or even if it were possible and they were to find a way, that fertilization is not going to actually happen when you put the sperm and the egg together. All right. But there's also what's called post-zygotic barriers. And so what that means is that you can have the matings that produce these hybrid individuals. However, they either don't survive very long after fertilization, they die shortly after fertilization, which means you got to the zygote and then it died. Or if the hybrid offspring do survive, such as the mule above, which is a cross between a horse and an ass, you get a donkey or a mule. They're infertile or they have reduced fertility, so they can't go on to make other mules. You have to keep crossing the previous two. So that's a post-zygotic barrier. Okay. So there was this guy by the name of Linnaeus, and he noticed when he went to scientific conventions that everyone was using a different name to describe the very same organism. So for example, when you think about the name for, let's just say, a house sparrow, well, some people would call it a house sparrow, and some people would call it a chickadee, and some people would, all these different names. And so when scientists were getting together, it was really hard to communicate with each other if everyone is calling the same species a different name. So Linnaeus said, listen, let us start using, a form of nomenclature of species so that we're all talking the same language when we're talking about species. And so this is what he came up with that is still actually used today. And so this is Linnaeus's organization of life. This is the Linnaean system of classification. And so it starts with domain at the very top. And if you remember, we have our eukaryota, which we're part of. And then we have the archaea, which are your extremophiles. Remember that? And then we have bacteria. So 3 basic domains. And then from domain, we go down to kingdom. And there are 4 kingdoms. So we've got Animalia, we have fungi, we have plantae, and we have protista. And of course, we are part of Animalia. And what we're going to end up doing is classifying this zebra here. And so zebra, of course, is part of Eukarya. Zebra is part of Animalia. You're going to understand later what that exactly means, what puts an organism in that classification. But then we go down to phylum. So it's domain, kingdom, phylum. Now we have, when we look at the zebra, the phylum of Chordata. There are other phylums, many other phylums. Then we go class. When we look at the zebra, the class is Mammalia. There are many other classes, right? Then we have order, which is Perissodactyla, family, Equidae, genus, Equus, and species, Quagga. And so when we speak of the zebra scientifically, we don't call it a zebra. We call it by its genus Equus and its species Quagga. So this is Equus quagga, okay? And then that way, no matter who you're speaking to around the world, even if they speak a completely different language, you're using the common language of science to discuss an organism, okay? So here's what I need you to remember. The order that the Linnaean system goes in: domain, kingdom, phylum, class, order, family, genus, species. Okay? And there's lots of mnemonics to kind of help you remember that. I also want to point out that the plural of genus and species, the plural of genus is genre. Okay? And so that's all great and wonderful that we have the Linnaean system of classification, and it tends to work fine. However, sometimes the biological species concept doesn't work, because remember, the species concept is built upon whether or not organisms can interbreed, right? Well, what happens when you're trying to classify an asexual species? It's not breeding at all, right? So asexual reproduction does not involve interbreeding, so the concept of reproductive isolation no longer meaningful. Or what about when you're classifying fossil species? You have no idea who they could breed with, right? So you end up using these differences in size and shape of fossil bones, which can't actually reveal whether there was this reproductive isolation between the individuals from which the bones came. And then determining when one species is changed into another. There's rarely this definitive moment marking the transition from one species to another. Like everyone's looking for the missing link, right, in humans, from monkey to human. Usually it doesn't happen. Usually you don't have this definitive one species that's right in the middle, you know? And then how do you classify what's called ring species? So ring species are two non-interbreeding populations that may be connected to each other by gene flow through this other population, right? So there's really no exact point where one species stops and the other begins. And then you have this other region where the biological species concept doesn't always work, and that's when you're trying to classify hybridized species. So in hybridization, the interbreeding of closely related species can sometimes occur, right? And produces the fertile offspring, suggesting that the borders between species are sometimes not really that clear cut. And so there's always this gray area. There's always this potential difference that doesn't, completely fit the definitions in biology, right? And that's the one thing that we have to remember in biology and science. Never say never, that you can have these outliers, right? That's just part of science. That's just part of biology. There will be an outlier, right? And so we have to basically deal with how do we classify these type of organisms, right? All right, so we're going to talk now some terminology. So I want to help you to lean on your Greek and your Latin. So allo means other, and patros means homeland. So let's talk about allopatric speciation. All right, so we're looking at how you actually get a completely different species. How does this happen? So allopatric speciation occurs when a geographic barrier causes one group of individuals in a population to be reproductively isolated from another group. So here we have our initial population here in step 1, and then we have this reproductive isolation. So suppose a river forms through the squirrel's habitat, separating these two populations. Now, because the squirrels can't cross the river, the two groups are now reproductively isolated. Over time, the populations on either side can diverge enough genetically that they're no longer actually able to interbreed, and then we end up with two separate species. That's allopatric speciation. Allos being other, patros being homeland. So we can see allopatric speciation when we look at the Galapagos Islands, right, due to allopatric speciation, there's 14 different species of finch have actually evolved in the Galapagos Islands, and only one species of finch is found on the nearest mainland. So we have the mainland finch, and then we have these 14 different species in the Galapagos Islands, allopatric speciation. But there's also something called sympatric speciation. So sympatric speciation results in the reproductive isolation of populations that coexist in the same area. So two scenarios can lead to this method of speciation. So one, let's take a look at the left-hand side here. So number one, chromosome duplication within a single species. So here we've got a cell division error can occur during mitosis, leading to this polyploid tissues, meaning 4N instead of 2N. So gametes produced by that tissue would be diploid 2N, right? Normally, your gamete, once it, you have the meiosis and division, ends up being haploid, right? But say we've got this one cell division error that occurred, and now when you go into meiosis, you end up with a diploid, right? Two diploids. So what can happen? So you can go on to fertilization, right? And now what happens is the individual with polyploid gametes can no longer interbreed with other individuals having normal gametes. Their offspring would have 3 sets of chromosomes which could not divide evenly during cell division, right? That can certainly happen. Or, and in that case, the offspring is sterile, right? They go on to live, however, their offspring are completely sterile. Or you can have a gamete with twice as many sets of chromosomes, can however produce offspring by fertilizing another gamete that also has twice as many sets of chromosomes. Or some organisms can do self-fertilization, so we have to remember that's possible too. And so now you have this whole new individual that has achieved instant reproductive isolation from the original population and therefore would be considered a new species. It has this massive number of chromosomes. So that's an example of sympatric speciation, how it can happen. When we look at another way that it can happen, so here we have combining chromosomes from 2 different species. So 2 plants from different species interbreed, they form this hybrid, okay? So take a look at our chromosomes that are here. So we have gamete 1, gamete 2, fertilizes, we get this hybrid individual. Now, the hybrid may not be able to interbreed with either parental species, right? There could be some barrier there. And then, due to a cell division error, the chromosome numbers, say, are doubled, right? Because you can see they're uneven to start with. The hybrid may, however, be able to propagate itself asexually, right? As many plants can do. Remember, we're not just talking humans here, we're talking other organisms that are on the planet. So many plants can propagate asexually. And so any future cell division error that leads to a chromosome doubling can then lead to individuals producing gametes that are fertile with each other. And so now you can end up with a new species. Okay, so that's sympatric speciation. So on the tree of life, there are many, many branches, right? Let's take a look at how to read an evolutionary tree, because when you first take a glance at an evolutionary tree, you can be misled by what you think you are reading, right? Unless it's being explained to you. So let's take a look at that. So evolutionary trees reveal the evolutionary history of a species and the sequence of speciation events that gave rise to them. So here, we're starting here with this common ancestor of mice, rats, humans, birds, fish, okay? So at this point, some speciation event occurred and the ancestral species split into 2 species. So here we have the split going into two species. And then there was a split again. Here we have the common ancestor of mice, rats, humans, and birds. We have the speciation. Here, fish had already split off. And then again, we go up, birds split off, and we have this common ancestor of mice, rats, and humans. We have speciation, humans split off, etc. Now, the evolutionary tree, this is important, evolutionary trees do not show which groups are more primitive or advanced. So it's not saying that humans came from rats. It's not saying that humans, came from birds. That's not what this tree is actually saying. So evolutionary trees do not show which groups are more primitive or advanced, just which groups are more closely related to other groups. Okay? So, in essence, the branches of an evolutionary tree can be spun around at the point which they split and still remain accurate. Okay? So, it's not saying that everything came from each of these. That's not what it's saying. It's just saying how they're more closely related. Because remember, the whole purpose of what we're trying to do with evolutionary trees, what we're trying to do with the Linnaeus system is we're just trying to find methods to classify organisms based on, it could be morphology, it could be anatomy, it could be the genomic code, now that we have that ability, right? And so these are just different ways to classify organisms. So evolutionary trees can be built in different ways, right? So for example, if we want to build the evolutionary tree based on comparative anatomy, well, here, let's just take a look at this comparison. So here we have the African lion and we have the spotted hyena. And so questions are asked. Does the individual possess retractable claws, right? The African lion does, spotted hyena does not. How many number of chambers are in the ear bone, right? Here we have 2. Does the penis have a bone? Here we have no, right? And so that's where we're actually getting this change, right? When we compare black bear, gray wolf, same questions are asked. We can see the answers are different. So we can see that there is a evolutionary split here between these two. And that's really what this evolutionary tree is trying to say when you are looking at it. And you have to know when you're looking at an evolutionary tree, well, what is it based upon? Is it based upon comparative anatomy? Okay, what are we comparing, right? Is it based on morphology, what they look like? What are we comparing in that? Is it based on the genomic data? Okay, what are we comparing? So you kind of have to be told what your evolutionary tree is actually basing its comparison on, so that when you're looking at the split here that's happening, you can have an understanding of where it's diverging between the two, okay? So this is really cool. You can get repeated evolution, and we've seen that before as species evolve. Remember, evolution is the change in allele frequency in a population over time. And so, but it can lead to some common forms. And here we're looking at the evolution of the wing. We can see it in the moth. The moth has a wing, and we can see a bat. A bat has a wing, right? So here's what's really interesting. When we take a look at these organisms, here we have the common shrew, we have the African golden mole, and we have the African elephant. Now, if we were to compare them based on morphology, what they look like, you would think that the common shrew and the African golden mole would have, be more similar, right? However, it turns out when we actually run the DNA sequence, it turns out the African golden mole, which resembles morphologically a shrew, but when we compare the DNA, the African golden mole has more DNA sequence comparisons that reveal that it is actually more closely related to an elephant, which is just mind-blowing, right? And so now that we have the ability to take a look at DNA sequences and compare them, we're starting to actually shift animals around on these evolutionary trees based on the genomic sequences. So more terminology that I want to tell you about. One is adaptive radiation. So what is adaptive radiation? So adaptive radiation occurs when a small number of species diversify into a larger number of species. And three phenomenon can actually trigger adaptive radiation. Number one, mass extinction events. So with their competition suddenly eliminated, after this mass extinction event, the remaining species that survive can now rapidly diversify. Okay, adaptive radiation. Or you can have colonization events. So moving to a new location with new resources, possibly fewer competitors, colonizers can rapidly diversify. Adaptive radiation. Or there could be evolutionary innovations, right? Change in this allele frequency over time that gives this new increased fitness. With that, a species can rapidly diversify. Okay, so that's adaptive radiation. So I do want to talk about extinctions because we are in the midst of an extinction event right now. So extinctions generally fall into two categories. One is mass extinctions, and that's the one that our brains naturally think about when we're thinking about extinction. So during a mass extinction, these large number of species become extinct over a short period of time due to extraordinary and sudden environmental change. Something horrific has happened, you lose all these species. But there's another type of extinction that's actually going on right now, and it's called background extinction. And so these extinctions occur at lower rates during times other than mass extinctions. So here we're looking at a time scale, percentage of families that became extinct, and the time over millions of years. And we can see that extinction of almost all the dinosaur species and 3/4 of all species on Earth occurred right about here, and we have time scale 0. This is where the dinosaurs went extinct. And then we have these other mass extinction events that have happened. There's one called the Great Dying that happened here a little over 200.5 million years ago. But look at this wave here, this graph, this portion of the graph. These are the background extinctions that are continuing to happen over time. And we have that happening right now. So when we take a look at this concept of after the origin of life, whatever happened that we ended up with this ancestral bacterium, the most commonly accepted tree of life suggests that after the origin of life, the following sequence of events possibly occurred. The bacteria arose from the first self-replicating, metabolizing cells. Then there was this split between the bacteria and a line that gave rise to the archaea and the eukarya. And then you had this fusion of bacterium and an archaea-like prokaryote that gave rise to the eukaryota. Remember, we talked about that before, which then split from the archaea line. And we have to remember that this classification is always changing as we learn more, right? But it's an attempt to try to classify and understand where different organisms fit in our classification. Now, I want to point this out to you because it's something that's really, really important, especially during a time where we're going into flu season, cold and flu season, in addition to COVID-19. But here, take a look at the bacteria here on this toothbrush bristle. This is actually magnified. This is one bristle of a toothbrush. It's magnified. All of this is bacteria. And so here we can see that bacteria can actually thrive in this huge range of habitats. And they have a biomass greater than that of all the plants and the animals on Earth. But remember, this is why you need to change your toothbrush roughly every 3-4 months. And certainly after you recover from a cold, flu, whatever, throw that toothbrush out, get yourself a new one. Otherwise, you may be just reinfecting yourself. Even though colds and flus are viruses, same thing. If we were to go in even closer on this toothbrush, we would see a whole viral environment, ecosystem that's happening on this toothbrush bristle. Right now, we can just see the bacterial ecosystem, but change your toothbrushes quite often. All right, so this is an example of our archaea. These are the extremophiles. They are a separate classification. Before, they were thought to be part of bacteria. They got their own classification, and of course, prior to that, we didn't even know they existed. And here we have an image of Geogemma barrosi, and it can live at 121 degrees centigrade inside this hydrothermal vent in the Pacific Ocean. And remember, in centigrade, water boils at 100 degrees, just to give you an idea of how hot that is. And that's the environment that this organism likes to live in. That is part of the archaea, our extremophiles. And that leads us to the eukaryota, which we know so well. These are the organisms we're most familiar with. We're part of the eukaryota, right? But so are slime moles, pink calla lilies, mushrooms, this antelope, and we're going to go further in the next lecture and look a little bit deeper at the diversity of life on Earth. So that ends this lecture. I'll see you in the next one. Bye.